continuing with the small intestines, we look more into how the digestive processes work in the small intestines. Now we know that the chyme that's found in the stomach is going to be sent into the small intestines. So it goes through that pyloric valve, or the pyloric sphincter for that matter, and goes into the duodenum. But what is it actually that's going into the duodenum? Well, it's partially digested proteins and partially digested carbohydrates, undigested lipids, undigested nucleic acids. Which means we've got to figure out, where do the carbohydrates initially start the digestion? Carbohydrates start the digestion in the oral cavity with the salivary amylase. Then where do the proteins start their digestion? They start their digestion in the stomach with pepsin. The small intestines will be the site where the lipids and the nucleic acids will initiate their chemical digestion. So different macromolecules, carbohydrates, proteins, lipids, nucleic acids, will initiate chemical digestion at different points of the GI tract. So what's going on here now is we have this slow delivery of this hypertonic chyme. So this is what's coming out of the stomach going into the small intestines. That needs to mix with bile, with enzymes, and with bicarbonate. Now all three of those are coming from a combination of the liver and pancreas. So the bile itself has to come from the liver via the gallbladder sometimes. If you don't have a gallbladder, maybe, then it comes straight from the liver. But the liver is what produces the bile. Gallbladder stores it and secretes it. Pancreas is doing a number of tons of enzymes being secreted there. But together, that's all being sent down into the duodenum via that major duodenal papilla, and it's mixed up. The mixing is the key. You have to make sure that you get all that chyme, all these enzymes, all this bile, and all this bicarbonate, all mixed together as evenly as possible. And that's done via segmentation. The segmentation in the small intestines is initiated by these intrinsic pacemaker cells. So it's an internal system inside of the actual small intestines. It's meant to mix and move all these components, that chyme, the bile, the enzymes, the bicarbonate, constantly just mixing it up and slowly moving it down towards the large intestines. The segmentation is not a fast-moving process. Its purpose is to mix everything up and slowly propel it on. Now, the intensity can be altered along the length of the small intestines. Your short and long reflexes play a part here. Remember, short reflexes, that's only going within the GI tract. Long reflexes, back up to the brain and down. But that's all based on feedback mechanisms. Also, it's going to wane in the late intestinal phase. Basically what that means is, as you get towards the end of processing your food, let's say you ate dinner and it's 4 hours later, 5 hours later, 6 hours later, however long it took to digest that food and move it through the intestines, once it's mostly gone, the stomach doesn't have any more coming in, that's the end of the intestinal phase. The whole thing starts to kind of slow down because you don't need all the enzymes. You don't need this constant segmentation because there's not as much chyme present, if really much at all. All right, just to refresh, we've seen this, but to refresh. External stimuli, so thinking about seeing, thinking about food, smelling food, is all coming through the brain. So this now is being sent from the brain down into the local intrinsic nerve plexus, which controls the actual small intestines. But then you also have the internal stimuli. This internal stimuli, this is the short reflex. It's things that respond within. So for example, if your chemoreceptors weren't picking up as many of a particular particle, that could tell your small intestines, hey, you might want to slow down the segmentation. We don't have as much here. or if the stretch receptors picked it up and tells it what to do. So it's based on internal regulation here. That long reflex went back to the brain. That's helping to control the overall processes as well. Your brain's telling it, hey, here's what's going on, here's what's happening. Helping control the movements within that, helping control that segmentation. So again, just to refresh what segmentation is as a visual, you're basically squeezing here, squeezing here, squeezing here. That comes over and it mixes that chyme up. And it keeps squeezing in between each bolus, each section. And it keeps continuously mixing and mixing and mixing until you have a nice homogeneous mixture, a nice even mixture. But that's slowly working its way down towards the large intestines. But there's also this second type of movement called peristalsis. Now, peristalsis is interesting. It doesn't happen initially in the small intestines. It's only happening towards the end of the process. Now, it's going to be initiated by motility. So this is happening now in the late intestinal phase. So when that segmentation is starting to slow down, peristalsis starts to pick up. Each peristaltic wave, remember, it's going to squeeze the tube and then run that squeeze all the way down. Squeeze the tube and then move down. Each peristaltic wave starts distal to the previous, so a little bit further down. This is the migrating motility complex. What the idea here is, you've already squeezed everything out of this point and started pushing it. Well, then start a little further down, and start further down, start further down. Each time you start further down, you're pushing all the remnants of that food, that meal. You're pushing the bacteria, the debris. Everything is being propelled along and out of the small intestines. So here we have this bolus of food, bacteria, chunks, whatever's there, and it's pushed down. The next one starts distal to that, a little bit further down, and pushes down. Next one starts a little distal and pushes down. The idea here is because once you've already pushed from this area, there shouldn't be anything left in that spot. But as you continue to run down the peristaltic wave, you might leave a few remnants behind. So you're always starting just a little bit beyond the previous peristaltic wave. Now, this whole process, this whole motility inside the small intestines is controlled by your local enteric neurons. So it's inside the actual small intestines. That's what's helping to coordinate and kind of keep everything in check and know what's happening at this point. You're going to have this cholinergic sensory neurons that are causing a stimulation and activation of the myenteric plexus. Now, we've mentioned the myenteric plexus before. Early on in this chapter, we're talking about the 4 different tunics. That myenteric plexus is inside of the actual lining, the actual walls of your GI tract. So what happens here is we're causing this circular muscle to contract proximally. And we're talking about the longitudinal muscle contracting distally. So the circular muscle contracts and squeezes and makes the lumen smaller. The longitudinal contracts and shortens the tube a little bit. This is helping to force the chyme along the tract, helping to move it and propel it down the small intestines. The goal is to get down to that ileocecal sphincter. The ileocecal sphincter is the end of the small intestines. It's the opening that goes into the large intestines. Now, what will happen here is you're going to have this gastroileal reflex. The gastroileal reflex is going to enhance or increase the segmentation in the ileum. When you increase the force and the segmentation there, it's helping to really push all that mixture, push that chyme through into the large intestines. Gastrin also is now going to help to increase motility of the ileum as well. Increased motility really gets things pushing and moving through, leaving the small intestines behind and going into the large intestines for the last section of your GI tract. Because large intestines is what will empty out of your body after that. Now, the ileocecal valve that we went through, it's a one-way valve. The flaps open up when you push through, but if anything comes backwards, the valves snap shut. So it's a one-way valve, just like we talked about in the heart, how those valves were one-way valves. So it's making sure that what goes into the large intestines, which is undigestible components, waste, bacteria, make sure all that stuff doesn't come back up into your small intestines. So those flaps are only going to open when you have the force from the ileum pushing into it. Otherwise, they remain closed, making sure that the two areas stay separated. That's the small intestines, some physiology of how you're moving the food through the small intestines.